This presentation will explain the poster Quantifying the Impact of Nitrogen and Phosphorus Levels on the Growth of Algae in Canyon Ferry Lake, and will be presented by introductory chemistry students Matt Cortner, Joy Denigan, and Katherine McEuen. This experiment analyzed nutrient concentrations in Canyon Ferry Lake, which is located on the Missouri River near Helena, Montana, and has been subject to significant algae blooms over the past years due to rising nitrogen and phosphorus concentrations. The presence of toxic algae blooms poses a serious threat for aquatic ecosystems and has caused great concern for the Montana Department of Environmental Quality, or DEQ. As students in an introductory chemistry class last fall, it was our goal to work in collaboration with the DEQ to determine the levels of nitrogen and phosphorus at which algae blooms occur in Canyon Ferry Lake. To perform this experiment, water samples were collected from Canyon Ferry and were later spiked with different concentrations of nitrogen and phosphorus. To begin the experiment, the DEQ ran tests to determine the initial concentration of nitrogen and phosphorus in the control samples, which were found to be 0.039 parts per million nitrogen and 0.016 parts per million phosphorus. The water samples were then spiked with sodium nitrate and dipotassium phosphate and were later incubated to promote algae growth. After the incubation period, the samples were analyzed using steady-state ultraviolet-visible spectroscopy, or UV-Vis, to quantitatively determine algae growth. Additional tests were performed by a senior-level capstone class, which used high-performance liquid chromatography to help quantify the algae growth in each sample. The implications and findings for this experiment regarding the management of Canyon Ferry Lake aquatic ecosystems will be discussed later in this presentation.
After the water samples were collected from Canyon Ferry, the spike team in the introductory chemistry class added the designated nitrogen and phosphorus levels based off the levels that were already present in Canyon Ferry determined by the DEQ. The nitrogen levels ranged from 0 to 5 parts per million, and the phosphorus levels added ranged from 0 to 1 parts per million. The DEQ predicted that in order to get significant results, the levels of nitrogen in parts per million would have to be higher than the levels of phosphorus. In Figure 2a, it shows the amounts of nitrogen and phosphorus being added to each beaker. Through each sample beaker, the results would be determined at what levels the nitrogen and phosphorus became problematic in Canyon Ferry. After the samples were spiked, each beaker was placed into a grow chamber where 120-volt lamps were kept on for 12 hours each day to ensure that the algae would continue to grow. Shaker tables were also placed inside the grow chamber in order to keep the beakers in motion. The incubation period was 2 weeks on the shaker tables to simulate a controlled situation of Canyon Ferry. At the end of the 2-week period, the largest algae growth that was found was the beaker that had 1.5 parts per million nitrogen and 0.2 parts per million phosphorus. In Figure 4, the graph shows the life cycle of the algae in the UV-Vis absorption spectrum after the 2-week period.
Results and discussion continued. After the incubation period, a matrix table was set up to visibly show and compare spiked samples. The horizontal rows contain phosphorus spikes, and the vertical rows contain the nitrogen spikes. The contour plot of Figure 5b shows the trends of chlorophyll concentrations within the matrix model measured with UV-Vis spectroscopy. The yellow and green contour bands show the areas within the matrix with the highest concentrations of chlorophyll. Figure 6 shows 2 different samples taken from the most visibly dense brown and green algae growth, both at 100 times magnification on a light microscope. Brown and green algae were the 2 most visibly different groups of algae growth. It is unknown whether the 2 samples are representative of the same species or not. Based on this experiment, the spikes with the most algae growth took place within the ranges of 1.0 to 2.5 parts per million nitrogen added, and 0.2 to 0.4 parts per million phosphorus added. Another major algal growth took place within the ranges of 0 to 0.5 parts per million nitrogen added, and 0.6 to 2 parts per million phosphorus added. With consideration to naturally occurring ranges of the 2 nutrients, The actual ranges are 1.039 to 2.539 parts per million nitrogen and 0.216 to 0.056 parts per million phosphorus, along with the ranges of 0.039 to 0.539 parts per million nitrogen and 0.616 to 2.016 parts per million phosphorus for optimal algal growth in Canyon Ferry.